So thank you for the introduction, Mary, and thanks for the opportunity to speak to all of you. Again, my name is Manoj. I'm actually a clinician scientist at the Ottawa Hospital. All my training, though, is basic science. Did my PhD and a couple of postdocs in the lab. In the last couple of years, I've been brainwashed by clinical epidemiologists, including some of them that I'll speak to you later on this afternoon. I think Laura Lynn's here somewhere as well. Basically, they've got me thinking about the way clinical trials are run and how rigor is demanded in that setting. However, when we're in the lab, we don't often think about methodological rigor and study design in the same way. And that might actually be one of the threats, we think, to preclinical studies and deciding, which agent we should actually proceed with through the translational pathway. So my learning objectives today are to define internal, external, construct validity. Don't worry if you've never heard of these before. Hopefully by the end of this presentation, you'll have a bit of a better understanding of what these are. And then also, I think more importantly, start to begin to understand how to implement these methods in the lab to address these validities. In terms of what I'm doing these days for research, I've got a lab, but I'm also very interested in how things are translated from bench to bedside. Sorry, I should have a little caveat here. What I'm going to talk about in about 15 to 20 minutes right now is something that we actually stretch over an entire day for BioCanRx trainees. So those of you in BioCanRx will be holding this workshop. We have already held it a couple of times. We'll be holding it again in a year or so as well. So this is very much a condensed higher level version of what we try to talk about over the course of a day. So thinking about bench to bedside translation, this is one example of a success story. So those of you that are in the OV field, you probably recognize this is TVEC. It's the first and only oncolytic virus that's FDA approved. It's used to treat melanoma. And what I've charted here, and this is a study that we did where we try to trace back every single thing that's been published on TVEC. And what we did was we mapped the efficacy. So across the bottom here, you have efficacy. And then each of those bars represents the efficacy of different studies. The top four bars there in blue are actually preclinical studies. So you can see the very first study back in 2003 looked at about 100% efficacy, basically. Every single tumor regressed completely. And then I want to draw your eye all the way down to the bottom there, the second last bar there, Anta-Baca 2015. That is the most rigorous clinical study that was conducted looking at TVEC. So this is a phase 3 clinical study looking at TVEC in patients with melanoma. You can see the efficacy is now about 10%. So you can see there's a dramatic drop between what we often do in the lab compared to what are the clinical realities when it's actually tested in a rigorous manner in clinical trials. So what are some of the reasons for this decrease in efficacy from 100% to 10%? We think there's many reasons, first of all, and some of them may actually have to do with the way trials are designed, clinical trials I'm talking about. But also I think we can do things in the lab that might help us better predict and better set up experiments to figure out what the truth is and in terms of efficacy or whatever outcome measure you're looking at in your lab. And this goes back to bias and validity. So what is bias and what is validity? So bias is, I'm sure all of you kind of have an idea in your mind of what bias is already, but one definition we can use is they are systematic errors in collecting or interpreting your data. So when you are conducting your experiment or you're interpreting your data, if bias is creeping into that, then it's going to result in a deviation of those results or any inferences you make from those results from the truth. So the corollary to bias is validity. And this is the extent to which a study's design, a study's execution, and its analysis actually prevents bias from creeping in. So if you address validities within your studies that you're doing at the bench, you may help prevent or reduce the risk of bias in your studies that you're conducting. So this is a nice little framework that we use to address validities in preclinical studies. There's 3 different types that I'll focus on for the remaining portion of this presentation. They're internal validity, external validity, and construct validity. So internal validity deals with design elements and Some design elements that you might have heard of include randomization and blinding, and these are methods and elements that are used to reduce the risk of bias. Briefly, external validity. This really simply is conducting replication studies, varying your experimental conditions. So you found it works in mice. Does it also work in rats? Does it also work in larger animals? You found that it works in 8 to 12-week-old C57 black 6 mice. Will it also work in an elderly population of mice as well? So varying your experimental conditions to address whether you get similar results when you change up your experimental plan a little bit. Thirdly, looking at construct validity, how well does the preclinical model, whatever you're using in the lab, actually reflect the clinical reality? So if you're looking at heart disease, for instance, how well does your model of LAD ligation actually mimic that patient that I see who walks into my hospital with a heart attack? So that's construct validity. We'll go into a little more detail for each of these. But before I do that, just to reiterate, why do these elements actually matter? Well, they reduce bias and thereby potentially increase the generalizability of results. And of course, through this workshop, we're really interested in generalizability from what we do in the lab in small animals, often to patients. So one slide just on external validity, again, just to give you a bit more time to think about this. And as I mentioned already, this is really thinking about generalizability across varying experimental conditions. So again, if you have used 8 to 12-week-old C57 black 6 mice, the mice that we usually use in our lab for different studies, can we still find the same results if we used a middle-aged rat or a middle-aged mouse? Or an elderly mouse as well. Similarly, if you're using all-female mice, could you also see the same results in male mice? Could you see the same results across different species, like rats and pigs, if what you're largely using is mice? And then finally, thinking a bit more higher level, could a different operator in your lab? So could someone who's trained or has a similar skill set to you in your lab go in with your protocol and replicate your results? And then something that's also been taking off over the last couple of years are a multi-center approach. So could a different laboratory with a similar skill set to your laboratory actually replicate the results that you found? And again, this might be another benchmark to say, well, maybe this is something we want to proceed from bench to bedside with. Moving on to construct validity. And again, as I mentioned, this is the extent to which an animal model corresponds to the clinical disease that it's actually meant to represent. The limitation is that, no matter what model you're using in a lab, it's never going to mimic the entire pathophysiology of whatever the disease of interest that it is that you're studying. So that's where you really have to start thinking critically. Well, what is the best model to use? What's the best animal to use? If you use mice like I do, because I'm just starting off and we don't have much of a budget and that's the cheapest animal to use, then that's what you're going to use just for feasibility. But does that actually mimic the typical adult patient that has sepsis that I see in my operating room? Probably not. The best disease induction. So if you're going to use MIAPAC2 to induce your tumors in your animals, is that the best tumor to mimic whatever downstream clinical condition you're actually thinking about? And then finally, what are the best outcomes to look at? Well, you may have used histology because that's what you do in your lab. However, is that the best outcome when you're thinking about a clinical trial, or would something more functional be more applicable? So these are things around construct validity that you have to start thinking critically about to address this particular validity. And then finally, internal validity. That's what I'm going to spend the remaining of this presentation on. And this is the extent that the design and conduct of a study prevents bias from creeping in. And again, bias is going to be a threat to the biological truth that you're actually seeking. So there's a few different terms that you might hear being used for internal validity. This includes methodological quality or bias prevention. So in other words, if you pay attention to internal validity, you are potentially reducing or preventing bias from creeping in, or the way we like to often phrase it is reducing your risk of bias as well. And for those of you taking notes, this is one to definitely to jot down and take a look at for yourself after this workshop. And this is the National Institutes of Health Preclinical Guidelines for Reporting of Preclinical Research. This was based on a consensus statement that was published in Nature 5 years ago now. And it was a group of preeminent scientists from across the world who got together and they said, well, there's a big problem in translation. There's a big problem in reproducibility. What can we do to address this? And they came up with this list of 7 items where they said every single paper and every single study really should try to address this. And if you haven't addressed this, you have to be transparent about reporting that you didn't address these issues. And I just want to highlight randomization and blinding at the top here because these are 2 aspects that deal directly with internal validity. So if you actually randomize your animals and you blind your experiment, you may reduce the risk of bias and you may increase the methodological quality of your particular study. So if you just Google NIH preclinical guidelines for reporting, you'll come right to this right away. Unfortunately, I don't have time to go into all the other 5 aspects there. This is what our workshop that we hold for BioCanRx actually goes into over the course of a day. But I will give you just a taste of what randomization and blinding are all about. So you may be sitting there saying, well, I use inbred mice. They're all the same. I use all male mice. They're all housed in the exact same way. They're all fed the exact same diet. Why does randomization matter? It shouldn't matter at all. And this is actually a really interesting story. This was the seminal work that really looked at this in a systematic manner. And it's around this agent called NYX059, which is an agent used in stroke. It's a antioxidant or free radical scavenger. So in the late 1990s and early 2000s, there were a number of labs studying NYX059 showing extremely promising results in reducing infarct volume and reducing all the signs and symptoms of stroke in preclinical models. Then this led to the SAINT-2 trial where they enrolled over 1,000 patients. They treated them with NYX059, and at the end of the trial, absolutely no effect. In fact, there were some small signals of harm even shown in those patients who had received NYX059. So the stroke community then went back and they said, well, what happened to all those really promising results that were published? They looked amazing. Why didn't this translate at all? So then one of our collaborators in the UK, Malcolm MacLeod, he went back and his group went back and they looked at all the preclinical studies where they had looked at NYX059. And this is just preclinical data you're looking at here. The gray bar that kind of goes across the top there is if you looked at all the studies together, what was the reduction in infarct volume? And it was about 50%. Then they sort of parsed it out into those studies that are randomized versus those studies that are not randomized. You can see that there's a clear difference. So those studies that are actually randomized, there is a much smaller effect size in reducing infarct volume. Perhaps if the clinicians who are translating this and the scientific teams that are translating it had just seen those studies that are randomized, they may have been less enthusiastic about enrolling 1,000 patients in a large-scale trial. Similarly, thinking about blinding, they saw a very similar effect, where, again, you have 50% reduction overall. That's a gray bar that kind of crosses all that. But those studies that had blinded, that's just under concealed there's a much smaller effect size compared to those studies that had not blinded their experiments. So again, this is empirical evidence, and there is many other studies similar to this in other fields now demonstrating a very similar effect. Randomization and blinding, they do matter in the lab. If you don't address these, you may get an inflation of your effect sizes or how efficacious your particular agent or intervention you're looking at actually is. So in terms of randomization, what is it and how do we actually implement it? Well, randomization, just put simply, it's the process of assigning participants to treatment groups. So in other words, it gives each of your mice, your participants in your trials that you're running in the lab, a random chance of being assigned to any of the study arms that you have in your experiments. This helps ensure balanced groups. It helps balance known and unknown confounders. And as well, if you actually operationalize randomization, it keeps your statistical test that you're actually running at the end of your experiment honest and valid as well, too. And then next, blinding. This is a slide put together by one of my collaborators, Caroline Ilka, who's an OV researcher in Ottawa. This is her definition. I like it. So blinding, it's basically hiding who got what. So hiding which mouse got which treatment, basically. And what this does is it reduces bias during the conduct of the experiment. So there's a little cartoon on the bottom there. Clearly, the undergraduate student is showing a bit more favoritism towards the control animals compared to those animals that are being exposed to whatever agent. And it also prevents bias from creeping in even to data analysis or data collection. And just as a thought experiment here, this is a sort of make-believe experiment here. We're looking at OV, where we have our mice and we're injecting them with tumors, either MIA-PACA2 or tumor plus fibroblasts, growing the tumors up to 1.2 to 1.4 centimeters. And then, once they've reached this size, they're injected with VSV delta 51 luciferase, so an OV. And then 72 hours later, we're assessing our tumor burden in these particular animals. So just thinking a little bit about randomization and blinding, and we'll just do a quick show of hands for each of these. So there's 4 steps here, basically. Just by show of hands, who would randomize in step 1, 2, 3, or 4? So who would do it in step 1? You can just raise your hands really quickly here. Who would randomize in step 1? Who would randomize in step 2? Who would randomize in step 3? And who would randomize in step 4? So, and who doesn't want to answer it? That'll be the most of you here. Anyways, it's basically you would probably randomize in step 1. So everyone's receiving the same thing in step 3 there, but you have 2 different groups that will be differentiated right at the very beginning there. So you'd probably randomize in step 1. And again, don't worry if this is all new to you. Again, this is meant to be sort of a high-level overview, and I'd encourage you to look at the Landis paper in Nature in 2012, as well as the NIH website, because it provides a bit more description around this as well, too. And in terms of blinding, rather than ask you, I'll just tell you that you probably should be blinding at every single step here. So you should know what you're injecting. You shouldn't know which mouse you're actually injecting that luciferase into in the end as well in step 3. And you shouldn't know what mouse received what in step 4 when you're assessing tumor burden as well too. And this is something we have operation in our lab, and I'd be happy to discuss that offline with those of you that are interested. Just another tool for those of you that are interested in randomization, how you might start to operationalize it immediately. You might have a statistician in your center who would be interested in helping you out, but even in the absence of that, you can use randomization.com. Here I've just filled out 6 different experimental groups. There's going to be 12 mice. That's where the 12 comes from in the bottom there. There's going to be 12 mice that we can treat in a day. And then when we press generate plan at the bottom there, it'll spit out a randomized list of 2 mice per each of those 6 groups, total of 12 mice in a day, and that's your randomization plan for that particular day. So going back, this is my last slide here, going back to my first slide there on TVEC and thinking about, well, what were some of the differences that actually occurred? Like, why do we have this 100% efficacy at the very first study that was published versus 10%, which was, again, the large-scale, rigorously designed clinical trial? When you go back to those preclinical studies, I think there's a number of issues which may have predicted this loss of efficacy or this drop in efficacy. First of all these preclinical studies were done in lymphoma models. So there was no melanoma model that was published until 2016. We actually don't even have the data there because the data was so incomplete that we couldn't actually put it up on this graph. So none of these were in a melanoma model, whereas the clinical indication TVEC is approved for is melanoma. Secondly, all these mice were young mice. So the typical sort of 8-week-old mice that we use in the lab because they're cheap. However, all the patients, the mean age for those patients in these clinical trials was much higher than that. They also often had other comorbidities as well, too. All these were young, healthy mice that were injected initially. Next, none of these studies at the very top were randomized or blinded. And finally, all of these were done in very sort of similar models. Again, that speaks to external validity, where they weren't really pushed to look at different models or different ways of introducing the intervention as well, too. So there's a number of issues. There's other issues as well, even the trials, like clinical trials I'm talking about that were designed, that may have contributed to this drop in efficacy as well, too. But again, just some food for thought for all of you. It's my last slide here. That's my email on the top there, Twitter right after that as well, too. It's a picture of our translational research group at the OHRI and the funders that help support my work. Thank you very much for your time.